our next speaker is Dr. Bit Fogley, who is a professor at UC Denver Institute Medical School. He got his PhD at ETH Chemistry and Applied Bioscience under Professor Pervusin, and he did his postdoc with AdBax, NIH, and followed by GE React Group. And he became an assistant professor 2017, CU Denver Institute Medical School, like I said, in the Department of Biochemistry and Molecular Genetics. And he's gonna talk about his really, really cool exact NOE method. Thank you.
Can you hear me and can you see the slideshow?
Yeah.
Can you also see the cursor?
Yep.
Okay, perfect. Yeah, thank you for letting me give this talk on a protocol that is actually in the transition to be supported by Cyano, which is not featured on nmrbox. But we also have an older version of our software on MATLAB, and I'm going to show a major part in MATLAB. Okay, I'd like to give a little bit a broader introduction to what we do, not only hands-on aspects, because our stuff is not that standard, and I really want to give you an idea of what our method is capable to do. So first of all, what is an ENOE? Or this is short for an exact NOE. As you all know, proteins or also nucleic acids are dynamic, and there's many probes for dynamics out there, and we want to add an additional one, or at least a more sophisticated form of a probe. So we measure exact NOEs, and in turn we can convert these exact NOEs into exact distances, and as such are really a nanoruler. These NOEs are between protons that are close in space. This means that we can really get through space measurements, not necessarily along covalent bonds. I have shown an example here. For example, we can measure the distances shown by these purple lines. As you see, these distances depend on time, so they're fluctuating, and our measurements are integrals over roughly maybe 10 or even 100 milliseconds. So as such, they really cover a very broad time window, for example, broader than just standard relaxation measurements. Then these distances in favorable cases have a very high accuracy. They can be better than 0.1 angstrom. So how do we use these distances? I guess you're all familiar to some degree with NOEs, conventional NOEs that are used as distance constraint. The use of those is shown in figure A to the left here. You measure a peak height of a cross peak in a nosy, and then you just convert this into a very generous upper distance limit. And you don't even use a lower distance limit. This is not strictly true for all programs, but in general, it's the case. This means that the lower distance limit for a spin pair is simply the van der Waals repulsion that is used in the program. Now, what we do, we replace this upper limit by a very tight upper limit. This is shown here. So for example, when we get a distance shown in black, we apply this very, very short, small tolerance up here. And we also use a lower limit. This is a little bit unusual, as I said, and this is also very tight. So as a matter of fact, we apply symmetric tolerances for lower and upper limits. Now, if we use these distance restraints, we gain a lot of information that is lost in conventional structure calculations. As I mentioned before, the NOE is a time average. So the fact that it is averaged already gives you some information on dynamics. But there's a second interesting aspect. Let's assume you measure two NOEs emerging from a proton, for example, proton number 1 here. And this proton is flexible, so it goes up and down here, spends some time close to proton 2 and some close to proton 3. Because the NOV has a nonlinear dependency on the distance, as a matter of fact, 1 over r to the 6th dependence. If you have just a short distance between two protons, just for a fraction of the time, this will boost your cross peak. You will observe a very strong cross peak. And in general, you would then say that these protons are close in space all of the time. So when you calculate an average structure, which is really the standard procedure, you would probably place this proton in the middle between proton 2 and 3, because you have strong NOEs to both of them. Now we started to play around with structures that allow multiple states. This means that only the average of these states, or the back predicted NOE of these states, on average have to agree with what we measure. For example, if you allow 2 states, you will end up with one position for proton 1 up here, close to proton 2, and the second one down here, close to proton 3. Of course, you can use this information in different ways. You can combine this with molecular dynamic simulations. That's what we started to do too. Or you can convert this to relaxation data. So the NOE is not strictly only linked to multi-state structure calculations the way I'm going to show it, but it's a first step. Okay. I'm going to give you a brief overview of the protocol that we apply. Okay, we measure noses. Here is the protein. We measure noses, and as opposed to the conventional way of nosy use, we measure a series of noses with different mixing times. This means that the diagonal peaks will decay over time, so they are pretty strong. Here for this 3-spin system, that's 24 millisecond, a very short mixing time for nosy. And you see, as we step through increasing times, these diagonal peaks decay. That's because part of the magnetization is passed on to neighboring spins, and this gives rise to the famous nosy cross peaks, shown in red here. And these peaks build up over time, at least until, well, within a specific time window. You cannot mix too long. Then we fit cross relaxation rates from these spectra. And to do that, we use both the decay of the diagonal peaks and the buildup of the cross peaks. This is also non-standard. In general, you just look at the cross peaks, but we use both. Combine this all in a way that I'm not going into. The programs will do that for you. You don't really have to know the details. And we will obtain these sigmas, the cross relaxation rates. They're directly proportional to the distance in this 1 over r to the 6th relationship. Once we have fitted all of those, we have to apply a quality check. This is very important. A nosy has hundreds or even thousands of peaks, and everyone who works with noses knows there is a lot of overlap, there is a lot of artifacts, and so on. So in our method, it's even more important than in standard methods that you really remove bad peaks. So if you have buildups that don't make sense, it's very likely that there's artifacts. So we apply this well, quality check and keep the good buildups. Then these buildups are then converted into distances. And with these distances, you can calculate the structure. Of course, you can just calculate an average structure as is done in conventional protocols. But as I mentioned before, we go further and usually calculate multi-state structures. You could also use these distances as restraints for molecular dynamic simulations or other applications. Now there is one important thing. the NOE is always contaminated with spin diffusion. This means that when you look at an NOE from proton 1 to 2, there is the direct flow that you want, but there's also indirect flows of magnetization. So proton 1 will give magnetization to a neighbor number 3, and this neighbor number 3 will pass it on to 2. This will boost the buildup in addition, and you don't want that. So we correct for that. So we use a PDB input here, estimate the spin diffusion, and apply corrections to the buildups here. You see the initial measured intensities here are a little bit reduced. Once you have a good structure shown here, you can use this new structure to provide a better estimate for your spin diffusion corrections, and you could do this iteratively. In practice, it's not really needed. If you have a good starting structure, you can do it once or maybe twice, that's it. We've been playing around with this method for about 10 years, and this is the gallery of the proteins we have studied. It's not complete, but near complete. So you see here the residue number, and I sorted the proteins in increasing sizes. You see that the largest sizes we were able to work with are about 160 residue proteins. As a matter of fact, we have 2 of those. Cyclophilin A is here, and currently we work on PIN1, which is a little bit more challenging because it has 2 domains and a flexible linker in between. For all of those, we calculated at least 2-state structures. You can also apply the method to much larger systems. However, you will not be able to calculate a structure, but you can look at very specific aspects. Here we measured distances only between methyl groups for a proteasome, which is 360 kilodalton in size. So this is huge. And it's probably one of the few methods that still work with such large proteins, maybe apart from just simple chemical shift assignment. We were able to get a couple of distances with reasonable precision. They are not really that precise anymore. You don't get an 0.1 angstrom precision, of course. And a second very interesting thing we started to investigate is the application of the same protocol to nucleic acids. This is our first test case. That's a 14-mer UUCG tetraloop. It's an RNA that is very well studied in NMR, so it was a great test case for us. Currently, we're also extending the methods to DNA and to more challenging RNA molecules. I just want to give you one single example, what kind of information is really contained in the ENOE. So this is our most recent study, even though it is one that started a very long time ago. When I was still at ETH, I had a grant for this, and at that time, Dean Strotz was my PhD student, but we just are in the process of finishing this project. I'd like to go back one more time. It's actually the smallest of the proteins we have studied. It's the WW domain of PIN1, and it's only 34 residues. This means that we get very excellent data and can, well, do really cool things, as I'm just going to show now. Okay, so this PIN1 protein is a cis-trans peptidyl prolyl isomerase, and the WW domain is the smaller domain. It's the one shown here, and it has a binding site for for different ligands with a specific sequence. And the larger domain, the PPIS domain over here, also has this binding site and is the catalytically active protein. Now, as I said, we're only focusing on the WW domain over here. We have picked different ligands that are known to have different effects on the interplay between these 2 domains. So one ligand here is shown to actually make the protein more compact. This means that a closed form where the 2 domains are form an interface is more populated. Then you have another ligand CDC. It's a derivative from CDC protein that has very complex interplay with PIN1. This is known to actually open up the protein, so the open states are more populated. You see a couple of chemical shift titrations here, and we start with black, which is the apoform. Now, if we titrate in either of these two ligands, we see changes in the binding site here. That's the loop shown in yellow. And you see that both ligands, one shown from going from black to yellow, and the other one going from black to blue, both of them have similar chemical shift changes upon titration. So this is residue 15, 18, 19, 21. They're all located in this loop. However, when you look at the part that forms the interface with the other domain, you see very different responses. These are the residues shown all down here, including about residue 28 to 34. You see that some of the residues even go in different directions of chemical shift changes. I have to say that these ligands don't directly bind to this site. So this is an allosteric interaction. And we wanted to investigate that with exact NOEs. I go through that very quickly. We have a lot of NOE buildups. We fit them, run it through our program. To give you an idea how dense our data is, this is one residue picked out. It's the best. It's the tryptophan 11. All of these lines here are NOEs that involve this tryptophan 11. We have more than 60 NOEs just involving this single residue. This tells you what an extreme density of information we have. If you think about it, the tryptophan has 4 degrees of freedom. If you just calculate the dihedral angles that, that are present in tryptophan. Okay, so we calculated structures and tested the violations of our NOEs when we increased the number of allowed states. So one state would be here. That would just be a regular structure, an average structure. We get these target functions shown here. Target functions are a measure for the violation of the NOEs in the structure. I mean, it's pretty much a sum over the difference between measured NOE and back predicted NOE squared. Now you see, when we allow more states, or number of conformers, as I have written here, this target function drops. This means that with 2 states or even 3 states, we can improve our agreement. From then on, it levels off. And so more structures are not really warranted in that case. And this is true for all 3 constructs we looked at, the apoform, and the bound forms with the, well, what we call the positive and the negative ligand or allosteric ligand. Here are the structures we calculated. So we just picked 2 state structures. Here in blue is the apoform. I superimpose about 10 2-state structures. Each structure then has a cyan and a blue state. And because every structure calculations produces the same 2 states, we can conclude that these states are not random, but they are in this data. Then we have something similar for the 2 bound states. Here we observe 2 structures, 2 states. And now we focus on the loop which forms the interface with the other domain. And we find something really interesting. If you look at isoleucine 28 here, which is actually the N-terminal part of this loop, we can plot the dihedral angles with the same color. For example, here, it's actually going, this is residue 29, excuse me, but it's the same as what you would observe for 28. You see 2 clusters. You see the cyan state and you see the blue state. Now we can do the same at the C-terminal part of this loop, which is residue 31, that's shown over here. Then you see that each blue state and each cyan state is still clustered. In other words, we observe a correlation throughout this loop. Now, when we add the first ligand, we actually see that these correlation is conserved. Just pay attention now to the gold color and the red color. Gold is not very well visible, as I'm realizing here, but it's over here. Red is here. If you look at the C-terminal part, you'll still see the separation. With other experiments that I'm not showing, we actually were able to show that the ligand just pushes the one state into the other one. So it's pretty much a repopulation of these two states. Now comes the very interesting finding. What happens with the negative ligand? Of course, you can do these plots again. You take the N-terminal here, you plot dihedral angles, and again, you see the black state here and the gray state here. So they're separated. That's not surprising. But now what happens if you look at the other side, or the C-terminal part of this loop? You still see two clusters, black and gray. However, when you compare the colors, you see that they are flipped. So this means that we actually observe an anti-correlation. So the two states here in residue 29 kind of cross over when you go through to 31. That's very interesting. If you now just hypothetically plot the PPIase domain onto the structure, then you see the following. The PPIase is shown in this service plot in pink, just the interface domain. And in ball representation, we still show the two states with the same colors. What do you see here? We have a steric clash for one of these states in apoform. This is the cyan one, would be inside the surface. So it's not possible, right? We observe the same with one of the ligands, and the yellow state would clash. But what do we see with the second ligand? We see that both the black and the gray state actually clash in the interface domain. And that's the explanation why this ligand pushes the populations towards the open state. The closed state is to some degree simply not possible anymore. So with that, we were actually able to show a very interesting allosteric mechanism. So just to show this in a very simple picture, we have either open closed states in the apoform. The purple shape here is the WW domain, and the blue one is the PPIase domain. Now what happens if we add the ligand? If we add the positive ligand in yellow, it binds here to the WW domain, and the interface segment is, well, it turns more binding competent, and you have more of this bound form or a closed form. Now what happens with the other one, with the negative ligand? It's the one in gray here. It binds, and it allows for two configurations in the interface. It's shown here and here, but as I have shown, there is this crossover or this anti-correlation. So this means either you reduce here the edged, the rectangular shape and boost the triangle, or the other way around. And both of them are really not binding competent with PPIase. And that's why we populate more of these open conformations. But this is just a great example of what you can do with ENOEs. I have to point out, when we calculate just normal single states, even using the ENOEs, all structures look pretty much the same. You would not see these effects I've shown here. Why? It's because this allosteric mechanism is really run through the deviation from average states, or in other words, through dynamics. This is actually the model that has been proposed by Dryden and Cooper in '84, the so-called dynamic allosteric model, as opposed to the mono model, which is actually shown here with the yellow ligand, where you pretty much do a conformational selection and just push this specific state in population. Okay, so that's now enough about what we can do, and now I'd like to move on to the software. Okay, as I have mentioned before, our newer versions of our program are run through Cyano. However, we started this whole software in MATLAB. And we have also produced in the end a very nice version. So our software is called INORA, which is short for exact NOE by relaxation matrix analysis. And what it does is it takes the intensities from a spectrum. At this point, or at least for MATLAB, it's either intensities in the tab format of NMR draw, NMR pipe, or you can just read in a simple file with intensities, hand edited. That's actually how I started many years ago before we improved the software. And then you get a lot of outputs. The most important ones, of course, the exact distances. This MATLAB program you can download from my website shown here, and it's also shared in the folder in nmrbox. Okay, now I'd like to explain a little bit how this one works. It basically integrates all the data steps or the data processing steps, starting from converting these spectral intensities into structural restraints. This whole program is organized in an object format. It's open source, of course, so you can modify it. It extracts the auto-relaxation rates of the decay, or the diagonal decays, and the cross-relaxation rate constants, mostly driven by the cross peaks, but that are also slightly impacted by the auto-relaxation rate. So that's why you have to fit everything together. Then it validates the data. We have some measures that you can use, or what I really recommend, you just look at each plot. You can plot them out. They're all in a folder, and just judge if these buildups or decays look good or not. Then we correct for the spin diffusion. As a matter of fact, we have two different approaches. Both of them have some advantages and disadvantages. We have graphical displays of the results. Then we generate directly the Cyana format of upper and lower distance restraints, the so-called UPL and LOL files. And because we have two different approaches to correct for spin diffusion, one of which is a little bit superior. It's basically a divide and conquer approach. And we can actually also apply this one for partially deuterated proteins. This is very helpful because when you move to larger proteins, you may use deuteration of your proton positions. And the most popular application these days is that you deuterate your entire protein and then you reprotonate specific methyl groups. And what we have found out is that your deuteration is never perfect. This may be typically 99% or something like that. This sounds like no problem, but in large systems, these residual 1% protons can boost your spin diffusion. This is very severe. I was surprised myself. And we can account for this with the simple approach. Okay. Then we have a manual. I'm not expecting you to read this. These are a few pages. I believe it's a very good one. It is in the folder that I uploaded onto nmrbox. You will also have it when you download the whole folder from my website. It really walks you through everything, and of course in much more detail than what I'm going to show you now. So the input. We have four types of inputs. First, obviously, you have to input your intensities, so your measured data. This can, as I have mentioned before, come either in a tab file, which you export from NMR draw, or it can come with simple intensities. I'm just showing here the tab file. I think most of you are probably familiar with that. So NMR draw uses just one single file that characterizes all the peaks you have. So when you detect these peaks, you will get this file here. Each peak will obtain an identifier number here. I think it's called an index. And then you get a lot of data that we are not really interested in right now, like peak positions in various units and so on. So there was no space to show it all. You will have one parameter that is the assignment, ASS, and it looks like that. We put the assignments of all 2- or 3-dimensional noses into here, and it has to follow this format here. So this means that the different dimensions are separated by this semicolon shown here. If you just detect peaks in just one nosy, we're not talking about nosy series right now, you will have one parameter that unfortunately is not shown here. I really regret it because it's a relevant one. It's the intensity parameter. It's called height here, and it will be among these dots. This is the peak intensity. That's what we are interested in. Now, if you have a nosy series, you don't want to assign all the noses. You just assign them in your easiest nosy. That's of course the one with the longest mixing time because you have the strongest peaks. And then there's a small script that is just provided in NMR pipe or NMR draw, where you can just go into all the other noses and it will read out the intensities at the same positions as the one you have assigned. And it will add up these parameters here, these A, so A underscore A0, 1, 2, and so on. And these are simply the relative intensities of these other noses. Of course, you start with the one that you have assigned. So this is of course always 1 because it's 1 time the height you obtained. And then the others will either decrease or increase depending on whether you have a cross peak or a diagonal peak. Obviously, this is a cross peak. So you have a decreasing intensity because we start with the longest mixing time and move to short mixing time. So it's a build-up. Then we also have an example of a diagonal peak, the third line here. So this goes the opposite. The longest mixing time has the weakest intensity and they increase with shorter mixing times. Okay, that's the most important file. But on top of that, you have to just provide some additional information. So first of all, you have to tell the program how the magnetization flows in the experiment. There's many different noses, and of course the program doesn't know which dimension is which. So you have to say, is the first assignment here is that the spin where the magnetization starts, or is it the one where it ends up? That's 2 parameters here. The one that tells you which is the origin of the spin. And then the second one is the one where you actually say, if you want to normalize your cross peak build up to the initial intensity of your spin of origin or the spin of destination. This would then be what you fit in the diagonal peak decay. So if you back predict the intensity of your diagonal peak at 0 mixing time, you have the magnetization that is just available when you start to mix. And we normalize our buildups with that. Okay, then in addition, you have to tell what the mixing time is, the mixing times, the tumbling time, and some other parameters that are less important. They are all set to reasonable default values. And finally, you have to choose the method which you use for calculation of the spin diffusion. And these also needs a couple of additional information like the magnetic field you use, or if you have stereo-specific assignments or not. So the MATLAB version is really easy. There is one setup file where you specify these 4 groups of inputs that I have shown. can just walk you through. The setup file has default values or default names, and you really have to change only a few. We just go through that briefly. So first of all, you have to give a name, because all your outputs will be written into a new folder, which will carry this name plus the date. Then you have to specify your initial PDB file that you use to estimate spin diffusion. Then you can choose which nomenclature convention you use. I think we have three. We have the new Siena convention, the old Siena convention, and the famous Markley convention from '98 that has been published by a lot of big names in NMR. Then you have to specify the name of your tab file. That's where your intensities are in. And again, you can specify your naming or nomenclature convention for that. So you can actually have different conventions for your PDB file and your tab file. Can be convenient sometimes. Okay, now I'm just scrolling down in this setup file. Here is the choice of the origin of the magnetization or of your spin. So is it the first entry in your file or is it the second? So in that case, it's the first. And here you choose if you want to normalize to the first or the second spin. Then you have to tell the program if you have a 2- or 3-dimensional nosy. Here we have a 3-dimensional nosy. Then we specify the mixing times in this vector here. I just see that we have an error. It looks to me as if we have, oh no, that's all good. It's this edged bracket. Okay, so these mixing times are given in units of seconds. So this would be a 20, 30, 40, 50, and 60 millisecond series. Then you can tell the program if your water has or how much D2O it contains. As you know, for the lock, you add some D2O. The thing is, when you start an NOV, for example, on an H-alpha proton and you transfer it to your amide, you will have a slightly reduced transfer because the deuterium in water exchange with the amides, and of course the deuterium you will not detect. So we recommend not to use too much D2O. We usually use 3%. That's really enough for, I think, for most spectrometers, so you won't lose your lock. This means that you have 97% protons, and well, the program will correct for that. Then you add the tumbling time here, the tau C. You can choose an average auto-relaxation rate for your diagonal peaks. This is helpful because the program will use the auto-relaxation rates of both spins that are involved in an NOE, but you can still get a pretty good result if you know only one of them. And then for the other value, it will just use whatever you believe is good. And we tested this a lot. It doesn't really matter that much. So in that case here, we put 5.3. That's probably about the average auto-relaxation we observed for the diagonal peaks. Okay, then these are default settings. I'm not talking about the stereo-specific assignment. I'm just saying we have tools that handle the fact that you often have methylene groups where you don't know the assignment, but you still want to use them for distance restraints. So you can read about that in the manual. Then here you choose the approach with which the correction for the spin diffusion should be calculated. In that case, it's the full matrix approach, which I recommend if you don't have some deuterated samples. It gives the superior results. Then you specify the B field. That was a 700 megahertz magnet in that case. And if you want to reduce the runtime a little bit, you don't calculate the spin diffusion through every atom present in a molecule. So you can say, what is the maximum distance from your two protons and and a spin diffusion through a third proton should still be considered. We found out that about 6.5 angstrom gives good results. I mean, it's that the error is probably in the per mil range, and the calculation is much faster than considering all protons. Okay, I'm running now the program. I have pre-recorded that for you before so that I'm not losing my PowerPoint presentation, but it's just like what you would do. You just run here. Okay, now it's pushing that. Okay, you see now in the command window below, there's a lot of messages, what's going on, and you already get outputs. You see a couple of decays from diagonal peaks, you see build-up peaks, you see a couple of correlation plots of distances that are extracted versus those in your initial PDB file. And finally, you get a map here of all your NOEs that were evaluated. Let me run this again because it's so fast. Okay, I hope you were able to get more of a glimpse of it. So here you see all the NOEs that we could evaluate directly plotted on the PDB structure that you input. So you get an idea of how dense your NOE network is. We also output a file that contains a Chimera script where you can just read it into Chimera and it will directly plot these distances. This is very nice to make figures for publications. Okay. So what outputs do you get in text format files? Because in the end you will use those most likely. You will get one file that is called sigma data out ISO. And this pretty much collects all the important data of your fits. It does not include everything. It's huge. It's typically about twice the width of an average computer screen. So I'm just showing you some. Of course, you have the atom names of the two protons or atoms involved in the NOE. Then you get the cross relaxation rates, the sigma. In that case, it's the average one because you were able to evaluate both cross peaks, the symmetry related cross peaks in a NOSI. Then you get the distances for the second one at least, it's 4.14 angstrom. Then you get the distances for each of the two cross peaks alone. You see it here. Then you see how much correction for spin diffusion has been applied to the cross relaxation rates here. Then you see the some data that came in from the original PDB file that's shown here. You see the distances in the PDB file. So interestingly, in that case, this distance is much shorter than what we measure. This means that the original PDB input, it was some kind of X-ray structure, I guess, was not very precise. And you get many more information here. Now, if you type in cyanaride.m, you will obtain the cyano format distance restraint files. So they are called UPL or LOL for those who work with cyana. They look like that. They just contain the names of the spins and the distance. In addition, you get a couple of parameters that tell you how you ended up with your exact distance. So for example, here it tells you if you had a unidirectional or bidirectional NOE, which means if you could evaluate only one of the cross peaks in your NOSI or both, and some other things. Then I mentioned that it's important to manually validate your data. So what I recommend is that you plot your buildups and your decays. That's achieved with these commands here. And the program will generate two folders, and each NOE gets its own PNG format file. If you have two NOEs, for the bidirectional NOE, where you can evaluate both cross peaks, both of the buildups will be plotted into the same file. That's the case here. That's a unidirectional one where couldn't do that. You can open them in any kind of program, for example, in this, what's the name, this viewer, and you can just scroll through them and kick out what you don't like. Okay. And now, currently we are transitioning to Cyana. And why? It's for the reasons that you have been discussing previously in the other talks. It is really this cumbersome handling of different file formats. So we decided to integrate this into Cyana, especially because our Inora program is actually then directly interfaced with the structure calculation. And obviously in Cyana, you can do this very efficiently. Okay, so what does it do? So Inora integrates into Cyana and has also many new features that are really nice. And this new version, this is really a quantum leap. So I expect much more people to use this new version. Why? It is very fast for interpretation and much more versatile in treatment of ENOE data than the implementation in MATLAB. Then we can also handle nucleic acids now, both RNA and DNA. We are currently dedicating a lot of time in exploring the possibilities here, but our first results are very promising. Then, as I said before, and the NOE data is typically so, the information of the data is so dense that you can do more than just an average structure calculation. So we can do multiple state ensemble structure calculations, which we have always done in Cyana. So it's very natural to integrate that. Of course, we can do that now here. Then we also have some tools that can automatically sort states or cluster states. I'm going to show this later, but you remember in my example, I showed structures that have two states. Now, every single structure calculation produces two conformations. And then if you take your maybe 20 best structures, you want to figure out if your two states are random or not. If they're not random, then each result should produce the same two states. For example, every run would give you this blue and cyan state. We used to do that manually, just figuring out, is this the case or not? But now you can just do that based on RMSD considerations. Then you can also create peak lists. These peak lists are actually in the format of XEasy and all programs that are related to that. These peak lists have intensities that you predict from a full matrix analysis of a PDB file. This can be helpful when you actually have a structure and you want to see what kind of nosy you actually expect. Then we have a new feature where we extract distance restraints from a single nosy. You remember I said that we measure a series of noses with different mixing times. And you can use our initial approach with just one nosy. Why? Because we fit the diagonal decay. And to fit the decay, you need at least 2 points. So we have a simplified approach. It's not as precise as the normal approach, but you can just measure one nosy, which saves a lot of time. I mean, typically it will save about 80% of the measurement time. And then we have a very detailed tutorial that is integrated into the Cyana Wiki webpage. We have a search window. You can enter comments you want to understand, and most of them are there. And this is still being updated by Dean Strotz, the guy mentioned here. Okay, it has been mentioned before that Catalina has some problems with migrating some software. So I'm at that too with Cyano. So on this computer, I cannot run Cyano. And as you know, we can run it on nmrbox either. But Cyano doesn't really have a graphical interface. So it's all text file based. So I'm just stepping through these files. Okay, so the first file you run, this is not NOE specific. This is whenever you run a classical structure calculation in Cyano, you have this init file that sets some very basic parameters. And some of the ENOE parameters are now integrated into that file. So first you read in the sequence of the protein that you study. This is always the case for any structure calculation. Then you do some other things I'm skipping. And then for the NOE analysis, you already enter your deuterium level in your H2O, because this is kind of a global parameter. Then you also set your tumbling time here. And if you want to use some estimates for your average auto-relaxation rates, you can set them here. So these here, over here, are the inputs. And then you run a second script, which pretty much copies the process that I've shown in MATLAB. So you read in mixing times here. It has a very similar form. You read in the magnetic field. You specify your auto-relaxation file that you write out. This is interesting because you can also read it. If you run the whole process a second time, instead of fitting your auto-relaxation rate again, you can read in a file that you manually prepare. This is very common because some of your decays are overlapped and you decide that you don't really want to use these diagonal peaks. So instead of using your fits, you can just use what you edited here. Then again, you choose which spin or which entry in your assignment is actually the spin where the magnetization originates from. So in that case here, it was actually the second entry. Then you tell to which spin you want to normalize your buildups. That's here. Then we skip this because this is all standard. Then here you define the PDB, the original structure you use to estimate your spin diffusion. And here you read in the intensities. Either you do that as in form of the tab file that you would obtain from NMR draw, and this then will be converted into the peak lists, which is really the XEasy format and therefore also the Cyano format. Or alternatively, you can skip all this and directly read in the peak file. That works as well. Then the major commands that really carry out the whole analysis are here. It's these ENOE commands. So you read in your init file that you have defined before. Then it reads in the spins that you want to use for normalization and the mixing time. And then the whole fit starts. So first, it will fit diagonal decays. As a matter of fact, just simple exponential decays to the diagonal peaks here. And it will produce plots. And let me scroll down in the script. Then it will write this, the obtained parameters into this row file. And then you will do the fit for the buildups. So this will be then the ENOE cross command. Then it will apply the spin diffusion correction, which is this block here. And then it will plot your buildups. You can actually now choose if you want to plot the buildups as you measure them, or if you want to plot the buildups after the intensities are corrected for the spin diffusion. You have many opportunities. You can also plot the fits through your measure peaks, also there, either before or after correction for spin diffusion. Then you will convert the cross relaxation rates into the distances. These are effectives here. And finally, you will generate the distance restraints file directly in Cyano format, which is the command here. And finally, we will write them out here. That's the UPL and LOL files. You will also get a small overview file with a couple of statistics. Okay. Now you're done and you are ready to run a multi-state structure calculation. So we use a file before the multi-state structure calculation that actually prepares the restraints for multi-state. This is nothing specific about NOEs. You can apply any kind of restraints you have, and they must be prepared for multi-state. It's because Cyano actually connects different states of the same proteins through a dummy linker. So Cyano is actually tricked into thinking that it handles just one big molecule. And because of that, you have to apply these restraints to each copy of itself. And that's happening in this prepare script here. There's not much you have to do except for choosing how many states you will use in the structure calculation. That's the parameter up here. And then you just give names of all the files you want to read in, the sequence file again here. Then here is an ACO file, that's angular restraints. That's typically if you want to use some telos outputs. And then further down here, that's the coupling constants. And if you want to use the noses, it will be further down here. I'm not showing that because it's really just changing the name. Okay, and now you're ready for the structure calculation. So you read back in these distance restraints you just generated, either from NOEs, which are the upper and lower limit restraints, or from other restraints. And this is almost the normal structure calculation file from Cyano that should look very familiar to you. So you read back in the restraints here. You can choose the weights, the relative weights of these restraints as you go through different stages of the structure calculation. So one means it's just used the normal way, but you can reduce the relative weight. So for example, the RDC here is not turned on in the very first step. Then, well, you calculate the structure here. This is all standard. You write out violations here. That would end up in the overview file. And in our specific case here, then we compare this to a reference structure, the output, so it calculates RMSDs. You don't have to do that if you don't want to. And then we also sort the states. So in that case here, we have two-state structure calculation. So it will try to find the same state in each calculation that we do. That would be the sort states PDB, and that's it. Okay. Now the output, Cyano gives you only text files with a few exceptions. This would be here, the plots of decays and buildups. This is very similar to the MATLAB version, so I don't really have to comment on that. And it also provides a plot of the target function if you run different numbers of states. So I have shown before that you can reduce violations when you allow for more than one state, which is typically happening when you have a dynamic protein. And then you have the output here. I have 3 examples. So that would be the output if you allow one state. In that case, we have such high quality of the data that it looks as if you have only one structure, but I think it's a superposition of probably 10 or 20. That's because this is the WW domain again that I have shown before with an enormous number of NOEs. Then you can run it with 2 states shown here. And you see that the sorting program was able to identify similar states. So you see that Yeah, typically here in the beta sheet region, you really see that each structure calculation produces one state further up here that is shown in cyan and the other one in blue. Then here is another example when you're on 3 states, also sorted by the states. Well, that's what I wanted to show in terms of software. I'm wondering now, okay, I think I've done about an hour. I think I'm not talking about NUS in our analysis. I think I just move on to questions. Just one last comment. We use NUS because we measure a series of noses, and this takes an enormous time. If you record 3D noses, and let's say you do 5 mixing times, you want to measure every nose for 48 hours, it takes you 10 days. With NUS, you can really reduce this measurement time. Is almost the same results, but I'm not going to talk about it, but I'd like to acknowledge some people that helped. I have to step through this now. Excuse me for that. I cannot speed that up. So a lot of the work has been done in Roland Riech's group, where I used to work before I moved to Denver, and especially Dean Strotz, who did his PhD, was involved in all aspects of this work. Then we worked closely with Peter Günther, who is the developer of Sienna, and we also had help from Frank Delaglio with the non-uniform sampling project that I didn't really go into now. Okay, so I think with that, I'm opening up for questions.
Thank you, Pete.
Stop share. I think I'm okay. I think I stopped sharing now.
So from the participants, any questions? Please unmute yourself and ask.
Okay, there is a chat here. I'm not sure if this is, no, that's old questions.
I don't see any questions from the chat yet.
Oh, okay. Actually, I have a question for relating to Ponderosa. How are the distances, the NOE distances used, or in this assignment process, does Ponderosa use spectral data, or is the input just the distance restraints that you would generate in Sienna?
Oh, so you presented and now you're asking. Okay, yeah, Ponderosa wraps up Cyan and Explore NIH. It has its own module, but it's less automated. So I made my own version called Udana inside. So if you want to provide noisy data as it is, and it's possible, also you can provide distance restraint as a UPL format too. So I mean, like your MATLAB, you generate UPL, then that can be used as input to Ponderosa.
Yeah, because an interesting prospect is to use the exact distances for assignment. The funny thing is, of course, in order to get exact distances, you need to run spin diffusion corrections. For that, you need at least an estimate of your structure. But still, we were playing around with exact distances that we didn't really correct for spin diffusion. They're still better than just conventional distance restraints. It's just something that may be interesting in the future.
Yeah, sure, we can try. So your MATLAB code is source code, right? It's not a binary format. If that is compiled, provide a binary format, I think that can be integrated inside of nmrbox platform as well, because it's also possible to install the runtime library in nmrbox, and then if that is executable, just using the library, I think it's possible.
Okay,
Yeah, the thing is,
sorry, I just want to interject one thing there. We do have a fully licensed copy of MATLAB installed on nmrbox months ago.
Oh, that's good.
So you would be able to take source code as is, not have to compile, and you can distribute that way.
Yeah, the thing is, we don't really want to distribute MATLAB too far. Actually, Dean doesn't want to, you know, because he spent a lot of time integrating this into Cyano. This is the future, clearly. But I'm, yeah, I mean, I would be interested in integrating that. The thing is, you know, we have two very different versions. And you know, it will divide potential users into two kind of clusters. But yeah, I may have to think about it. Yeah.
Yeah, it should be cleared between Peter Günther and nmrbox fellows, I think.
Oh yeah, absolutely. But I mean, the MATLAB version is not really. This is we did this without Peter. Peter came in, you know, with the integration into Cyano. And actually, I mean, Dean is now in his group. Yeah. But so that there is no. So Cyano will not be integrated into nmrbox, or are there?
It's a work in progress. I won't get into the details at this point, but it's something we're interested in and we're trying to make happen.
Yeah, okay, that would be great, of course, because then we could just completely switch to Cyano. It's much more helpful. The thing is, I got a lot of emails of people who were interested in running the MATLAB version, but there was hardly any outcome. I think they just got disencouraged because most of them worked with even prior versions, not the one I have shown. And it was very complicated for them to run it, even though we thought it's kind of user-friendly. But if you're not an expert, it has to be very, very easily accessible. And that's why I believe that with the integration into Cyano, it's really a quantum leap. I feel like Cyano is not necessarily more user-friendly, but there's so many people that work with Cyano and they already know the structure and most of the commands that it's just easier. So you don't have to make an extra effort. It's just really naturally there.
Okay, thank you. Any other questions?
Can I just ask about, have you done any cross-validation? Because you showed these structures, the quality of the structures improved. But I'm just wondering if you've cross-validated that with some data which you haven't used in the calculation.
Yes. We did a lot of cross-validations in very different forms, also for our older tests. So one test that we do routinely now is the jackknife test, where we remove typically 10 or 20% of the NOEs, calculate the structure with the other 80 or 90%, and then we repeat this in a way that each NOE is excluded once in one of these 10 or 20% classes. Then it just back calculate the target functions from these distances that were not restrained. And then we check, for example, if this cross-validation target function also drops from 1 to 2 states. With less complete data, the 2-state calculations are about at the limit, which means that then the jackknife doesn't really show an improvement. But we also have a lot of other cross-validations, especially for what I've shown here. The thing with these 2 states, we did relaxation analysis, we did CEST, you see it in CEST, and we tested this population shift. I didn't really go into detail, but when you add the positive ligand, then one state is populated by about 80%. We tested that, yeah. But it's cross-validation is always a little bit tricky, I have to say. And this is not a general problem of NOEs. I mean, this is general with structures. Of course, yeah, I don't know if you're alluding to the statistics that are usually done, like Ramachandran violations. With that, it's interesting. We have a larger violation because then each state is evaluated in a separate way. And I think the Ramachandran statistics is really based on average structures and probably cross-validated like that or generated in that way. And our goal is actually to find the excursions from these average structures. And that's why they kind of always go under the radar of classical cross-validations.
Yeah, I was more thinking about a completely different data set. For example, if the fit to the RDCs improve.
Oh, yeah.
When the ENOEs.
Yes, we did that not with the example I've shown, but my major test case was GB3 protein. It was 56 residues, and we did a lot. Among others, we used a lot of RDCs. For example, we calculated structures just based on NOEs and back predicted the RDCs. And we see that the RDC back prediction also improves when you add more states. We also did the same with J-couplings. We did it with cross-correlated relaxation in the current case, actually. Yeah, we did a lot. The thing is, you can do that for a specific case, but I'm pretty sure each is different because it really depends on the topology. It depends on how many NOEs you can get. It depends on how the quality is. And yeah, I mean, I recommend to do the jackknife procedure because you can always do that without additional data.
Thank you.
Thanks again, Pete.